Hello dear students, welcome to this lecture series in botany. Today we will discuss about an important topic which is discovery, nomenclature and characteristics of enzymes. Enzymes are the biological molecules that catalyze specific biochemical reactions. They are active under mild conditions of temperature and pressure found in the cell. They are usually single or multiple chain proteins that act as biological catalysts that catalyze intracellular and extracellular biochemical reactions. On the basis of their composition, enzymes are of two types: simple enzymes and conjugated enzymes. The simple enzymes are entirely made up of proteins, while as the conjugated enzymes consist of a protein part and a non-protein part. The protein part is called apoenzyme and the non-protein part is called prosthetic group, coenzyme or cofactor. The complete enzyme including prosthetic group, coenzyme or cofactor is called holoenzyme. Apoenzyme consists of one or more polypeptide chains of tens to hundreds of amino acids. The prosthetic group is tightly bound to the enzyme and is usually a much smaller inorganic portion of an enzyme. It's attached to the protein part of an enzyme by covalent bonds and is essential for its catalytic activity. The simple prosthetic groups are metal ions such as copper, zinc, manganese and molybdenum. Some prosthetic groups may also be organic compounds which include cytochromes, flavonoids, pyridox or biotenol phosphate, heme or biotin. These groups are bound to active site of many enzymes that participate in catalysis. Coenzyme or cofactor is not tightly bound to the enzyme. The former is an organic molecule and the latter is inorganic in nature. Coenzymes serve as carriers of various types of chemical groups. NAD functions as carrier of electrons in oxidation-reduction reactions. Several other coenzymes are involved in the transfer of a variety of additional chemical groups, for example, carboxyl groups and acyl groups. Cofactors are essential for the catalytic activity of an enzyme. Some of the examples of cofactors are chlorine, copper, iron, zinc, cobalt, potassium and calcium ions. There are also some special types of enzymes. These are isozymes and extremozymes. Isozymes are the multiple molecular forms of a single enzyme which is normally formed under normal conditions. They can differ in various physical and chemical properties and their intracellular localization. Extremozymes are the enzymes which are active under extreme conditions of pH, temperature and pressure. These are well adapted to tolerate such harsh conditions. Enzymes have the ability to increase the reaction rate or biocatalysis without being destroyed or changed in the process. Because of this, one enzyme molecule can theoretically change an infinite amount of substrate if given an infinite amount of time. Increasing the amount of enzyme decreases the time required for completing the reaction. The enzymes enhance reaction rates by a factor of 10⁵ to 10⁻17 times than that of an uncatalyzed reaction and facilitate the attainment of chemical equilibrium fast. Enzymes function by lowering the free energy of the reaction's transition state. They are effective in very small amounts. They are extremely unstable and are inactivated or denatured by high temperature or very alkaline or acidic conditions. For an enzyme to work, it must have access to its substrate, the material upon which it exerts its action. If no substrate is available to enzyme, the enzyme performs no function. The substances on which an enzyme exerts its action are called reactants, and the substances produced at the end of the reaction are called products. Enzymes control all biochemical reactions in every living thing, from viruses to man. Breathing, digestion, heart action, formation of body tissues, movement of muscles, and many more processes depend on enzymes. So, without enzymes, life is not possible. More than 1 lakh different types of enzymes have been identified, most of which are colorless solid, soluble in water or dilute solutions. But some are blue, green, or greenish brown. The discovery of enzymes can be traced back to 19th century. The term enzyme was introduced by Kohn in 1878. It is taken from the Greek word zyme, meaning leaven. Enzymes are the principles found in leaven. The first observation was carried out on vegetable and animal cell-free extracts. Berzelius in 1836 coined the term catalysis, which in Greek means to discover. Claude Bernard in 1848 discovered the role of pancreatic juices and described the action of pancreatin, an albuminous substance. In 1864, Bichamp observed that the enzyme zymase is involved in the transformation of saccharose into glucose. Raphael Dubois in 1886 discovered luciferase, the enzyme responsible for bioluminescence. Later in 1896, several oxidative enzymes were discovered by Gabriel Bertrand. He introduced the concept of cofactor. In 1897, Hill discovered that maltase catalyzes a reversible reaction. Edward Buchner in 1897 showed that a yeast extract completely lacking cells could just as well stimulate alcoholic fermentation. He hypothesized that yeast cells ferment sugars not inside the cell but by secreting proteins into their environment, which is the actual mechanism. He was awarded Nobel Prize in Chemistry in 1907 for this valuable work. In 1926, James Batchelor Sumner demonstrated that urease could be isolated and crystallized. He showed by chemical tests that it was a protein. This was the first experimental proof that an enzyme is a protein. In 1937, he succeeded in isolating and crystallizing a second enzyme, catalase, and devised a general crystallization method for enzymes, and also showed that all enzymes are proteins. By similar methods, John Howard Northrop also obtained other crystalline enzymes by this time, starting with pepsin in 1929. Wendell Meredith Stanley is also known for the preparation of enzymes and virus proteins in a pure form. In 1946, James Batchelor Sumner shared the Nobel Prize in Chemistry with John Howard Northrop and Wendell Meredith Stanley for the brilliant work on enzymes. Nomenclature of enzymes. Many enzymes have usually been named by adding the suffix "ase" to the name of their substrate or the catalytic reaction. Some enzymes have been named in an arbitrary way. Some of the examples are trypsin, pepsin, chymotrypsin, rennin, and emulsin. The earlier system of nomenclature has been modified in 1992 by International Union of Biochemistry and Molecular Biology. Nomenclature system of enzymes, commonly known as enzyme commission numbers. It developed certain rules for nomenclature and classification of enzymes. The system divided enzymes into 6 classes, each with subclasses and sub-subclasses on the basis of the reaction catalyzed. According to standard nomenclature, each enzyme is assigned a 4-digit code number following the abbreviation EC and a systematic name which identifies the reaction catalyzed by it. The 6 classes of enzymes are oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. Oxidoreductases. Oxidoreductases bring about redox reactions or oxidation reduction reactions involving transfer of hydrogen or oxygen atoms, hydride ions or electrons between the molecules, that is from one molecule to another molecule. Some of the common examples of oxidoreductases are alcohol dehydrogenase, cytochrome oxidase, and heme oxygenase. Transferases. Transferases catalyze the transfer of functional groups from one substrate to another. Thus, they are involved in group transfer reactions. Transferases catalyze the transfer of an atom other than oxygen or hydrogen, or a group of atoms like acyl, alkyl, or glycosyl. Some of the common examples of transferases are hexokinase, glutathione S transferase, and methionine synthase. Hydrolases. Hydrolase catalyzes the hydrolytic cleavage of a chemical bond such as carbon-oxygen, carbon-nitrogen, carbon-carbon, and some other bonds, including phosphoric anhydride bonds. The main physiological function of the enzyme is the reaction with water as the acceptor. Some of the common examples of hydrolases are alkaline phosphatase, uracil DNA glycosylase, and exonuclease. Liases. Liases catalyze the cleavage of various chemical bonds such as carbon-nitrogen, carbon-oxygen, carbon-carbon, carbon-sulfur, and others by reactions other than hydrolysis and oxidation. Thus, they remove a group of atoms from the substrate and lead to the formation of a new double bond or a new ring structure. Liases require only one substrate for the unidirectional reaction, but 2 substrates for the reverse reaction. Some of the common examples of liases are fumarase, isocitrate liase, and pyruvate decarboxylase. Isomerases. Isomerase catalyzes the interconversion or structural arrangement of optical, positional, or geometric isomers. These enzymes catalyze structural or geometric changes within one molecule. In certain cases, the intermolecular oxidoreduction reaction is involved. They are classified as isomerases and not oxidoreductases because the donor and the acceptor groups are present in the same molecule. They may also contain firmly bound NAD or NADP. Some of the common examples of isomerases are inositol 3-phosphate synthase, phosphoglycomutase, and type 2 topoisomerase. Ligases. The word ligase has been taken from the Latin word ligare, which means to bind or to glue together. It catalyzes the joining of 2 large molecules to form new covalent bond coupled with the hydrolysis of a small dependent chemical group. For example, a pyrophosphate bond in ATP, GTP, or a similar compound is hydrolyzed by ligase. The enzyme links together 2 compounds and catalyzes the joining of carbon-nitrogen, carbon-sulfur, carbon-oxygen, and carbon-carbon bonds. Some of the common examples of ligases are DNA ligase, pyruvate carboxylase, and succinyl-CoA synthetase. Characteristics of enzymes. Enzymes can easily be identified by the characteristics possessed by them. The various characteristic features of enzymes are specificity, reversibility of action, temperature sensitivity, pH sensitivity, high catalytic rate, colloidal properties, molecular weight, chemical nature, and inhibition by poisons. First, the specificity. Enzymes are highly specific in the type of reaction they catalyze and the choice of reactants they act upon. A particular enzyme can catalyze only a particular type of reaction. Each enzyme acts on a single substrate or a small group of closely related substrates. The presence of specific functional groups adjacent to the bond to be cleaved determines the specificity of the enzyme. The active sites of enzymes are highly specific centers composed of varying number and sequence of amino acids and a particular binding site which complexes only with a specific substrate in a lock and key process. Reversibility of action. The enzyme catalyzed reactions are reversible as well as irreversible. In the presence of an appropriate enzyme, the conversion of substrate to products is accelerated without altering the equilibrium between the reactants and products. The enzymes enhance only the rate at which the chemical equilibrium is established and doesn't change the direction of reaction. They can accelerate the reaction in either direction. Many enzymes which are required for the synthesis of starch and sucrose are responsible for their breakdown also. However, by removing the products of enzymatic reaction as quickly as they are formed, the reversion can be checked. The enzyme must accelerate both the forward and backward reactions equally. On the other hand, some enzymes carry out irreversible reactions only in one direction. For example, hydrolytic enzymes catalyzing cleavage of bonds. In fact, large molecules such as fats, proteins, starch, and nucleic acids are synthesized by one enzyme and degraded by the other. Temperature sensitivity. The enzymes are sensitive to heat and hence are thermolabile. They function best at optimum temperature range of 25 degrees Celsius to 40 degrees Celsius. Both low as well as high temperatures are unfavorable for enzymatic activity. Their activity decreases with decrease as well as increase in temperature. Enzymatic reactions occur slowly at 0 degrees Celsius because of the lower level of the molecular kinetic energy, which limits substrate-enzyme collision and attainment of the transition state. With increase in temperature, both these events are favored. With every 10-degree rise in temperature, the reaction rate shows enhancement. Most enzymes are inactivated or denatured by temperature above 60 degrees Celsius. It's because majority of the enzymes are proteinaceous in nature. Early denaturation by heat is observed in enzymes present in moist conditions. However, the enzymes in drier or dehydrated conditions are quite stable. Denaturation disrupts the secondary structure of an enzyme and it loses its active site and hence the catalytic power. However, some enzymes are resistant to high temperature. pH sensitivity. In the catalytic site of an enzyme, the state of ionization of amino acid residues is pH dependent. Since the specific states of ionization of these residues determine the enzyme's catalytic activity, therefore, enzyme's activity is also pH dependent. Different enzymes function at different pH ranges. There is a restrictive pH range called optimum pH, which remains constant for each enzyme and is its characteristic property. The activity of an enzyme decreases with increase or decrease in optimum pH. pH changes also modify the ionic substrates. For example, if a COO group of an aspartate residue of an enzyme is used to bind a positively charged substrate, protonation would reduce the force of attraction and decreases the affinity of the enzyme for this substrate. Most enzymes show maximal activity around neutral pH, but there are many exceptions. Catalytic properties. Enzymes increase reaction rate by a factor of 10⁵ to 10⁻17 times than that of an uncatalyzed reaction. Enzymes are active in extremely small amounts. For example, one molecule of catalase can effectively catalyze conversion of 50 lakh molecules of hydrogen peroxide into water and oxygen. The enzymes remain unaffected at the end of the reaction. It also doesn't alter the position of chemical equilibrium of a reversible reaction, but helps in its quick establishment. Weak interactions, hydrogen bonds, hydrophobic and ionic interactions between substrate and enzyme provide a significant part of the energy used for reaction rate enhancement. Carbonic anhydrase is one of the fastest enzymes known. It catalyzes the hydration of carbon dioxide and transfers it from tissues into the blood and then to the alveolar air. Each enzyme molecule can hydrate 10⁵ molecules of carbon dioxide per second. Colloidal properties of enzymes. Enzymes are large proteinaceous substances of very high molecular weight. They have the size in the range of colloidal particles, that is 0.001 micrometer to 0.1 micrometer in diameter. Their colloidal nature provides large surface area for reaction to take place. Enzymes display all the colloidal properties. They don't pass through a collodion, but can pass through filter paper. This helps us in their separation from true solution. The enzymes, being colloidal particles, have positive or negative charges. Molecular weight. Catalase has a molecular weight of 2,50,000 Daltons and is one of the largest enzymes. On the other side, peroxidase, one of the smallest enzymes, has a molecular weight of 40,000 Daltons. Thus, enzymes are larger than usual simple organic molecules. Chemical nature of enzymes. Enzymes are mostly proteinaceous in nature, except a small group of catalytic RNA and DNA molecules. Their catalytic activity depends on the integrity of their native protein conformation. If an enzyme is dissociated into its subunits or is denatured, it loses its catalytic activity. Breaking down an enzyme into its component amino acids always destroys its activity completely. Thus, the primary, secondary, tertiary and quaternary structures of proteinaceous enzymes are absolutely important for their catalytic activity. Inhibition by poison. The activity of an enzyme is altered by many foreign substances that combine with it in a way that influences its chemical substrate binding property and turnover number. Substances or agents that reduce the activity of an enzyme or inactivate it in this way are called as inhibitors or enzyme poisons. Inhibitor binds to enzyme either reversibly or irreversibly. Thus, the inhibitors are of two types: reversible inhibitors and irreversible inhibitors. Reversible inhibitors can easily be removed from the enzyme because they bind reversibly to the enzyme by non-covalent interactions. There are three main types of reversible inhibition of enzymes. These are competitive reversible inhibition. A competitive inhibitor binds at and blocks the binding site of substrate on the enzyme. Most of them resemble the substrate and combine with the enzyme to form an enzyme inhibitor complex, but don't lead to the catalysis. By adding more substrate, its combination with enzyme is favored. Uncompetitive reversible inhibition. An uncompetitive inhibitor binds only to site distinct from substrate active site and binds only to enzyme substrate complex. Ultimately, it leads to no reaction. Mixed reversible inhibition. A mixed reversible inhibitor binds either to enzyme substrate complex or directly to the enzyme. It also binds to a site which is distinct from substrate binding site. Irreversible inhibitors. These are the inhibitors which bind covalently with an enzyme and destroy a functional group on it that is essential for its catalytic activity. Some inhibitors form a particular stable non-covalent association with the enzyme, but the formation of a covalent bond between an irreversible inhibitor and an enzyme is common. So dear students, I hope that you have understood today's lecture well. See you next time with another lecture. Till then, goodbye.